The overall goal of this procedure is to objectively measure the responses of individual cells by quantifying specific fluorescent marker intensities from microscope images. This is accomplished by first subjecting adherent tissue culture cells to siRNA-mediated gene knockdown. In the second step, the cells are fluorescently stained and then imaged for several markers of interest. In the final step, the expression of the markers within specific subcellular regions are algorithmically defined for individual cells. Ultimately, the cellular responses to biological cues from gene knockdown can be analyzed by measuring the fluorescent expression levels of the markers of interest and their subcellular translocation.
Though this procedure provides insight into the regulation of G1 cell cycle transit in response to siRNAs, it can also be applied to studies generating fluorescent cell images, studying nuclear transport and protein homeostasis. Demonstrating the procedure will be Daniel Wetterskog and Ka-Kei Ho, fellow postdocs from my laboratory.
Begin by dispensing 70 microliters of 200 nanomolar siRNA diluted in siRNA buffer into the appropriate wells of a sterile 96-well plate in a sterile tissue culture hood. Then add 105 microliters of transfection lipid diluted in 40 volumes of serum-free DMEM medium into each well of siRNA. Mix the plate by gentle vibration at room temperature, and then after 10 minutes, subdivide the resulting 175 microliters into 3 50 microliter replicates per target into an opaque tissue culture treated 96